Hello, and welcome to the program. My name is Sam Ankerson. I'm the Executive Director of the Bailey Matthews National Shell Museum, and we're here to enjoy Dr. José Leal's talk, Micromollusks, the Allure of Small Size. And very glad that you've joined us. And we'll bring José on screen in just a few minutes. But before we do, I hoped to give you a brief update on how the museum is doing since Hurricane Ian. Many of the people registered for this program are close followers of the Shell Museum, but for those who are not, the Hurricane Ian in last September delivered a major impact to the museum, and with our monthly lecture series, we'd like to give an update on how things are coming. So with that in mind, I have just a few slides and that we'll take you through, and then look forward to introducing José and getting on with the talk. This is a picture actually that José took just a couple of weeks ago on May 1st of the museum at sunrise. And it was taken the day after the museum closed. So there was actually a period of time from February 1st through the end of April when the museum was open. In those months following the storm, there was a lot of work in cleaning out the museum and gutting it. But from February through April, we were open to visitors, and we were able to continue being open to visitors through the end of April. And but then reconstruction work started in mid-April and really began to pick up, and it became necessary for us to close again. And that reconstruction work is well underway. We have several crews per day in the museum every day. Here we see some of the work going on the ground level, where the aquarium exhibits are, where the museum store is in the lobby. And what they're doing here is reframing and putting in insulation, getting ready to put in drywall and new ceilings. We are going to be making some changes for those familiar with the museum to some of the layout on the ground level, namely changing the configuration of the store to increase the amount of shopping space in there, and then looking at potentially some aesthetic changes to the aquarium exhibits. No fundamental change to the aquarium exhibits, same tanks, same species of animals being exhibited, but some of the labeling and some of the aesthetics in there, we're looking at making some updates to. One of the major impacts of the storm on the museum, in addition to all the flooding on the ground level, was a major hole that was torn in the roof. And in addition to the hole, there was the compounding problem of the truss, the structure of the roof being heavily damaged in many places. So this is work that has now been completed, that happened in April, to rebuild a lot of that structure on which the roof rests. And that's been completed. All that carpentry work's been done, and that means that roofers can come in and do a permanent repair on the roof, which is scheduled for the first week of June. Back down to the ground level, some other work besides the framing and the insulation and prepping for the drywall. The giant Pacific octopus aquarium, and there's a picture of this on the left. Fortunately, the floodwaters of the storm, one thing they did not do a great amount of damage to were the tanks themselves in the aquariums, with the exception of the giant Pacific octopus tank, which started rusting, and just the amount of salt water that it was kind of marinating in was having an effect. So on the left is a crew that came in and sanded it and treated it and repainted it, and it's now in good shape moving forward, and has been blessed by an engineer. Electrical rewiring that's been going on for weeks now, and that's happening throughout the ground level. And the last piece that remains there is to place some of the major pieces, panels and transfer switches and things like that. But the electrical work is proceeding along. We expect work, this kind of reconstruction work is going to continue for perhaps another 8 months. Our goal is to reopen the museum in phases, with the aquarium exhibits opening first, we hope by the end of the year, and then the Great Hall of Shells and the second floor to happen after that. We hope during season of 2024. All the contractors are lined up, and work is going well. We expect the total cost of repairs and exhibit redesigns that we're looking at to exceed $3 million. And the museum has been, of course, fundraising since the storm, and there has been an insurance payout, and we are working with Small Business Administration on a loan, and working with FEMA, and looking to put all of it together. But it's a major endeavor for the small museum, and one that's going to require attention and support moving forward. So, but glad to say that it's underway. And while it was great being open for those 3 months, and we'd love to be open now, the good news is that there's just so much work going on that it isn't practical. You can keep up with the museum's progress through our website and through social media updates on Instagram and Facebook. Apart from that, from reconstruction and all of that, we are continuing to offer programs such as the one you're here for right now. A couple of upcoming online lectures are listed right here. These are not yet posted on our website, available to register for, but they will be soon. July 13th, Dr. Noreen Young from the Bernice, I'm going to mispronounce it, but Pauhai Bishop Museum in Hawaii will be giving a talk on Hawaiian land snails. And August 17th, Dr. John Pfeiffer from the Smithsonian will give a talk on freshwater mussels. Our own José Leal will be doing a talk in October on cowries, and we have other lectures to announce as well. But these, like this program and previous online lectures, these will be free. And as soon as they're available to register for online, we will send it out by email and our newsletter and all of our usual channels. A new field education program we debuted in January, partially in response to the hurricane. It's a series of biodiversity walks and land snail searches throughout Lee County. And the next one is coming up next week in Fort Myers. And this program is also free, and you can register through our website by going to the education tab and clicking on that. And we'll be announcing another series of about 6 of these biodiversity walks through the summer and fall that will again start to include Sanibel and Captiva, as those areas are now more accessible than they were. And excuse me, right here are some photographs from an exhibit that the museum organized, community photographs of Hurricane Ian and its aftermath, in which we invited community members from any hurricane impacted area to submit their own photographs of their experience or anything that was meaningful to them about the storm or what came after. And we wound up receiving about 150 photographs that we exhibited at the museum when we were open. But now that we're closed, this exhibit is available online, again, through the museum's website, shellmuseum.org. Click on special exhibitions. Right now, there are about 145 posted, along with statements from the photographers, and more are being added all the time. So this exhibit will continue to grow, will continue to be online at the museum's website, and we are currently looking for actually another public venue in the area that might be interested in taking the photographs themselves while the museum is closed. So I recommend going on our website and checking out the exhibit. It's quite powerful and also uplifting too. Not all the photographs are of damage and mayhem. There's a lot of hope in there too. So with that, I will before bringing on José, just want to mention that tonight's talk will be recorded, and as all of our online lectures are, and available for free on the museum's website. And so it usually takes us less than a week to get it up on the website. And so we'll encourage you to view it later, share it later, and check out any of the 20 other lectures that are now available online through the museum's website. If you have questions on José's talk tonight, please use the chat function and type in your questions, and after his talk we'll come back as we are right now and do some Q&A. And with that, I'll introduce my friend José Leal, who's been the curator here at the museum since 1996. He is now not only the curator but also the science director. He was director of the museum from 1996 through 2013. Nobody knows this place better. His resume, the list of accolades and honors is too long to read them all. But he's an honorary faculty member at the University of Miami and also Florida Gulf Coast University. He's involved in just a myriad number of ways in publication and research, and but among those is he's the editor of the Nautilus, which is I think the oldest malacological journal publishing and sort of housed here at the museum because of José. And has held leadership positions in Conchologists of America and American Malacological Society and many other entities. So with that, I'll introduce our good friend and colleague and science director and curator José Leal. And he's also, I would say, he's humanity's answer to the chatbot. So
thank you very much.
Enjoy the talk.
Share my screen now. I hope you all can see that. I thank you so much, Sam, for your very nice introduction, and thank you everyone for being here tonight. Why micromollusks? Well, to me, micromollusks are both a passion and a necessity. A passion, you'll see why today, I hope, with this talk. And a necessity because every time you deal with the biodiversity of mollusks in a given area, let's say Sanibel Island or Key Biscayne, if you're counting, if you're listing the species that are in that area, micromollusks will be a big chunk of it, and we'll see a little more about that as we go. I have a couple of items here, and then we'll dive into the the not so small world of small mollusks. But again, why micromollusks? Well, basically they're there, and most people don't realize that. You'll be maybe walking on a sandy beach, let's say, the north end of Sanibel, Blind Pass or Bowman's Beach, and you'll be walking that sand, and without knowing, you'll be maybe stepping on literally hundreds or thousands of little shells. I have an example that I like to cite when we talk about this subject, is that I did a little snorkeling with some Mexican colleagues back on the Caribbean coast of Mexico, and I saw a sand that looks really rich near a coral reef, and I picked up what ended up being about 2 pounds of sand. I brought that sand back to Sanibel, to the museum. This was early 2000s, and washed the sand, and gradually over weekends at home, I sorted that sand and ended up with 120 different species of shells out of 2 pounds of sand. That gives you an idea of how diverse they are, and how also how ubiquitous they are. What are micromollusks? Well, it's just micromollusks is just a semantic concept. I mean, we consider micromollusks to be any mollusks with shells that are smaller than equal or smaller than 5 millimeters. You may find elsewhere, different size limits to that, but by and large, we consider micromollusks to be equal or smaller than 5 millimeters. I'll be showing you some species today that may be a little larger than that, but I hope you forgive me for that. But hopefully never larger than 12 millimeters, which is about a half an inch. The lure of the allure, as the title said, of micromollusks or microshells, is that some of those shells, they may be as attractive and colorful as those of the large counterparts, especially in the gastropod class. So and they have life processes as complex as their larger relatives. As a matter of fact, some of the very small micromollusks, especially the microgastropods and microbivalves, they have to face a higher water viscosity, because as animals get smaller, they perceive the water in a different way than larger animals do. The water to them becomes more viscous, almost like honey, and they have to suck that in. So they have special resources, special systems that help with that also. And the final reason has to deal with biodiversity. And the fact that micromollusks will, as I just said earlier, micromollusks will form a very large fraction of the mollusk biodiversity in any given area. And I'll be showing you a very good example of work done by colleagues a few years back. Forward when moving next. Just to give you an idea of size, I have here a cent coin, a penny, and a little ruler. A penny is about 19 millimeters. This is our established size limit for micromollusks. You can see here, just for comparison, a sewing needle with the needle eye and a straight pin with the pinhead. And as a matter of fact, you'll see next that a lot of the shells I'll be showing you are of the size of this pinhead, or even smaller, which is pretty cool. 5 millimeters are about a 1/5 of an inch, 0.2 of an inch. And you'll get an idea next. Again, here is our good old penny and its size, 19 millimeters. And here an iconic local Sanibel micromollusk, the obese dove snail, Parvanachus obesa from Sanibel, right at the size limit for a micromollusk, about 5 millimeters. But that's not as small as it gets. We have shells here that are gastropod shells that are much smaller, such as the small callus vitrinella, a big name for a very little shell. And keep in mind that all those images here are at scale. So this is the one that has this, it's about the size of a pinhead. It's one of the smallest species on Sanibel Island. This particular shell here measures about 3/4 of a millimeter. Oops, go back. Sorry. 3/4 of a millimeter, or 3/100ths of an inch, which is pretty small. They are the small callus vitrinella is a member of the Thornid family, which together with the Teinostomatidae have those flattish shells that are very, very common here on the island. We have in excess of 20 different species. All those little guys are from the islands of Sanibel and Captiva, and I'll talk a little more about a few of them later. The largest one in the family here is the bow vitrinella that measures about 8 millimeters in diameter. And we usually measure the diameter by picking up the longest dimension this way here, as if you're measuring a circle. And the smallest one in this shot is the small callus vitrinella with, 3/4 of a millimeter in size. This is a question that I'm sure you have. How do you distinguish a true micromollusk, let's say a little gastropod that never grows beyond their little small size? How do you distinguish that from a juvenile of a larger species? And as you know, shell growth begins very small. Shells begin very small. And as the body of the animal grows, the animal mantle, the animal itself, will make more and more shell. In gastropods, that will be coiling around, the shaft of the shell. Here are 3 examples of juveniles, very small individuals of large local gastropods. A horse conch, all of them very well known, a horse conch, a nutmeg, and the infamous or famous Junonia. This is a little Junonia here. The scale line is in millimeters. This is not necessarily the size of a micromollusk, but still a very small size. The nutmeg here is about, I would say, about between 3 and 4 millimeters in size. Well, how do you distinguish those guys from true micromollusks, such as the 3 species here on the right? A couple of things. It's not, set in stone. There's not a magical recipe for that. It may not work in all cases. That's my point now. But it does work in many cases when the protoconch, which is the remnant of the larval shell in a gastropod, when the protoconch is relatively large in relation to this total shell size, that indicates that it could be a juvenile of a large species. Look at how much smaller than the true micromollusks, how much smaller are the protoconchs, which are the remnants of the larval shell. Another little hint, another little clue, and again, it may not work in all cases, but it may work in some cases, is the presence of a thickened outer lip. This area here outside of the opening, outside of the aperture of the gastropod shell, is thickened in adult micromollusks, or in this case, microgastropods. So just keep that in mind for future reference. I mentioned that a study that was done on a given area showed that the number of micromollusks, the diversity of micromollusks, it can be really high. And it's just an example, but that example may be extrapolated, for other areas. This study was published in 2002, so it's 21 years old. And since they did that, there were other groups of scientists that kind of replicated the same kind of study for different islands, different parts of the world. But this was a group done by Philippe Boucher and his colleagues from the Paris Museum, the National Museum of Natural History in France. They found that in a small area of the island of the main island of New Caledonia in the Southwest Pacific, repeated visits, repeated collecting, collected about 2,738 species in this very small area, of which 2,581 were mollusks with shells. Remember, there are mollusks, sea slugs, or in little cephalopods, and in other classes that may not have a shell. So out of 2,738 2,581 were shelled mollusks. And I'll show you in the next slide the proportion of those shelled micromollusks, which is 2,581. What proportion had shells, were micromollusks? I just give you an idea here. This is the island, the main island of New Caledonia, which is a French territory. It's called, the French call it an overseas territory of France in the Southwest Pacific that measures about 300 miles in length. That's the area of Kumak on the southwest coast of New Caledonia. And this is a map that I took out of Google Earth. And on that photo, on that aerial photo, I will superimpose the area they studied and pictures of the little stars here showing 42 stations. So this is the coastline here. Some of the stations where scuba diving were done, collecting were done by scuba diving. And just to give you one idea of the size of this area, the length of this area here is about 20 kilometers or 12.4 miles with those 42 collecting stations. Just to give you one idea, Sanibel Island measures about 27 kilometers in length, or about 17 miles. And if I superimpose Sanibel, this is a, like a cutout of Sanibel Island, superimpose on that area they studied, gives you an idea more or less of the area for those of you who know Sanibel. So in an area that's smaller than Sanibel Island, they found those many, in excess of 2,000 species. And of those 2,581 shelled mollusks, about 864 were equal or smaller than 4.1 millimeters. So that's a heck of a lot of microgastropods. And that pattern can be seen also in different parts of the tropical world. It may not be the same in temperate areas, but studies done in tropical areas have shown more or less the same kind of proportion, about 33% of very, very small species. We have a similar ratio here for Sanibel Island with a lot of local species that are very hard to identify or impossible to identify. I want to show you, talk a little about collecting. This is a picture that my good friend Susan Hewitt, who I hope is in the audience tonight, I asked for a picture of her collecting, and she does use this method that's very unique, and I know other people do that, but she collects micromollusks by literally diving in the sand. She wears knee pads, elbow pads, and also a loop visor. I'll show you a picture of that later. And she finds the shells this way. Susan has made a huge contribution to our local collection, and she's published a lot on some of the Caribbean islands. And she's also one of the curators for iNaturalist, one true citizen scientist. Most of us, when collecting micromollusks, we don't do that. That's a very special gift she has of being able to see them in the field, I mean, right there. Most will collect by picking up mollusk-rich, shell-rich sand, taking it home or taking it to the lab, washing that, and using simple pieces of equipment like brushes, tweezers. This is the magnifying loupe, like the one that Susan uses. And then proceeding to pick up the shells out of that sand, out of that sediment. Keep in mind that it's important to wash everything in fresh water. You don't want that salt to stay there because it can be damaging to the shells in the future. If you're really becoming serious, if you turn out to become an enthusiast, I mean, a real enthusiast, you may want to spend a little more and buy a little microscope. There are outfits now, mail order, I mean, online order, if you will, where you can get a microscope like that for anywhere between $300 and $400, which is not a lot of money when you think of it. So that helps sorting the material and will help even more when we begin trying to identify your shells using online resources of good old shell books. This is a picture of my hand, took another day, like 2 days ago. And this is one of many, many vials that are waiting to be sorted, that were donated to the museum by Herb Chapin, who collected here in the late 90s and the first few years of the 2000s. So he collected sand, good sediment out of beach access number 3 on Sanibel, and sorted all the shells. He didn't, in many cases, he didn't separate the species, put them in vials with all the collected information. This one was collected in May 1999, which is about 24 years ago, at low tide. And just waiting for that moment when we have free time to be sorted and incorporated into the collection. But that shows you basically the result of sorting good sand for micromollusk shells. Here in the museum, we got lucky in the last few years, let's say within the last 5-6 years, we received a couple of grants that help us, set up a really nice photo lab, also with our own institutional support. I mean, we couldn't do that if the museum administration didn't like the idea, obviously. So the grants were one IMLS, Institute of the Museum and Library Services grant for the digital imaging project. Many of you have heard of that, where we set out to photograph about 4,000 specimens, 4,000 lots of our collection. And then the second grant, the National Science Foundation grant under the Thematic Collection Network program, titled Mollusks of the Eastern Seaboard. So in that lab, we did a lot of imagery. We did about 4,000 different lots. And I will show you a little of the equipment we use for larger shells. We use a camera, a old Nikon D800 with macro lenses on a copy stand, and for the small ones, for the micros, we use this really nice setup with a very powerful so-called long-distance microscope made in the US by Infinity, just a Max long-distance microscope. And then we use for process of focal stacking. Some of you may be familiar with that, so that we get all the shell in focus. We use a Cognizant stack shot focus stacking system. There's a picture of one of our digital imaging project supporters. James Kelly was one of the photographers we hired back in 2019 actually, and he was our leading project photographer. And a lot of the pictures you'll see today of micromollusks next were taken by James, and we're really grateful for all the hard work he put into the project. I dabbled myself during my Ph.D. candidate years, and you'll see next what I did. I dabbled a lot with scanning electron microscopy, and that's a process that gives you very, very high definition images of very small mollusks. This is a West Indian Alvania, actually from Brazil in this case, that I shot this with many, many other ones under the University of Miami scanning electron microscope. This is a picture of me here, who knows when, way back into the geological past. Working, I did work as a technician there, and had the go ahead to use the machinery to do my own work in off hours. So I with that support from Dr. Pat Blackwelder, who was the head of the electron microscopy lab at the University of Miami back then. And she just passed away a couple of weeks ago. So Pat was super supportive, and thanks to that, I could include in my PhD thesis, which was then later published as a book, I could include a huge number of images. If I have that book, a book has about 418 pages, and if you're interested, just email me and I'll give you a link to download the book free of cost. It's open access now. So I have that book has about I think a little less than 30 of those plates, and most of them, if not all of them, are of true micromollusks, but also juvenile shells of larger species. Yeah, those, as you can see, the scanning electron microscope photos do not have color. The process used doesn't see color, and also doesn't let you see the transparent nature of some of the shells. But it's a super medium for enhancing detail, for allowing us to see detail. There are also fossils of micromollusks, and I was speaking with Dr. Harry Lee from Jacksonville, who many of you know, citizen scientist of the higher caliber. I was talking with Harry at the last Florida Malacologist meeting in St. Pete, and Harry said, well, I know you're going to talk about micromollusks, why don't you show some of my shots of fossil micromollusks? And he sent me quite a few, and I made a selection. Here are a few photos taken by Harry and his colleagues at the Florida Museum of Natural History, now named the Florida Museum only, and of some fossil micros from the Pinecrest Formation in Central Florida. Pretty cool, aren't they? Look at the shapes. It's pretty impressive. You know, all those things are, and again, those are little fossils that if you go to a fossil pit, if you're not collecting specifically for micros, you'll never get to see them. This talk is mostly about shells. We're not talking a lot about the animals, their systems, their lifestyles and all. But I just want to show a couple of pictures that I'm very happy with that I took of some dove snails from Sunnyvale Island. This is the fat dove snail. And you can see those dove snails, some of the species, the very small ones. And this one, again, is that iconic 5mm species. They can actually swim. They can adhere their feet under the surface, the water surface. So kind of sailing under the surface tension layer, the air-water interface. They're so small, they don't fall. They just hang in there, which is pretty cute. Again, 1mm is 4/100th of an inch. Another local micromollusk, another very small one, it's the lunar dove snail. The shell has those beautiful little patterns that look like brown crescent moons, if you will. That's a false sparse dove snail. It's probably an unnamed species that looks very much like a species called the sparse dove snail. You can see the little guy here. This one is one of the, I'm breaking my own rule here. It's larger than 5mm. You can see the little guy in two different views here, and you can see the tentacles, you can see eyes, the siphon, just like, your large marine snail. A shell also of a deeper water dove snail off Key West. So this is actually a shell that was collected 30 miles of Sand Key, which sits just outside of Key West. It doesn't have the colorful patterns you see in the shallow water species, so it's a species that lives in deeper water. Sam mentioned today that we're going to be refurbishing part of the Great Hollow Shells, actually a lot of it. And in the old Great Hollow Shells, we had a micromollusk exhibit that was a very small exhibit with a few samples of micromollusks and the photos of them. We'll be revitalizing or renovating this exhibit in the new setup. So keep your eyes peeled for the new micromollusk exhibit section that will be under the extremes exhibit that we're planning on doing. We're pretty cool. You see that exhibit had a backdrop of a scanning electron microscope image of a local Sunnyvale shell that I took many, many years ago of this unknown species, at least unknown to me at this point, that I believe belongs in the genus Pergolina. Here is the same photo now, so you can see. The scale line here is 100 micrometers. This scale line of 100 micrometers equals 1/10 of a millimeter. So this shell is probably the smallest shell we'll be seeing here today. This shell is probably about 0.5 millimeter in size. And also here, also two other species from Sunnyvale, a Cryoteris in a juvenile stage, and the dubious Serith, Finella dubia, also a commonly found micromollusk on Sunnyvale Island. Again, one of the very small shells from Sunnyvale. I've shown this species before today in a scanning electron micrograph format. The West Indian Alvania oberiana. Not a very good photo. I took those photos myself. They're not as good as James' photos. And here I have the scanning electron micrograph version of the same species, so you can compare, not very good resolution and outstanding resolution, but you will lose the color, you lose the translucent aspect of the shell. I'll be showing you some like relatively cool looking. Actually, I find them to be very cool looking. I hope you agree with me at the end. Some of the local Sunnyvale species, some not so local. And this is the Catisby riso, Schwarziella catisbiana, and it's a shell that never grows longer than 4.6 millimeters, so it's still under the limit for a true micromollusk. Another species of the same family, Rhizoid, that's the smooth rhizo. It's a very cool looking shell. It's totally smooth. And if you shine a light through it, you see the shell is transparent, it's translucent, actually not totally transparent, but translucent. And the smooth rhizo may measure 5.1 millimeters in size. Next, the gem cyclostream, also from Sunnyvale, that never reaches beyond about 3.5 millimeters. Pretty cool looking. It's related to trochids and turbans. You can tell that if you remember the looks of a chestnut turban. It kind of reminds you of that. Sits in the same general superfamily as the chestnut turban. There are three left-handed species or at least three left-handed species on Sunnyvale. One of them, I don't need to tell you, it's a lightning whelk. But then we have a couple of triphoras. And this is the modest triphora that may reach about 5.5 millimeters in size. Notice the left-handed aperture here in the same fashion as the lightning whelk, only a much smaller aperture, but also left-handed. And then again, we have a very special species that you find only in mangrove areas. Actually, this shell and a few other ones in the same lot were collected inside red mangrove bark by a colleague and former museum volunteer, Dr. Jim Scatterday. It's left-handed also. It's called the left-handed Melampus, and not an easy species to find. You really have to be looking for it to be able to collect that. Not from Sunnyvale, but also from Florida, collected off Fort Lauderdale and Broward, the exquisite false dial. And I know it's a favorite species of our finance director and jack of all trades, Ryan Powell. And I put this shell here as a tribute to Ryan, who I know is watching somewhere else. So the exquisite false dials, Pyrrholaxis centrifuga, has a shell in which the whorls don't touch each other. The adjacent whorls do not touch, the other ones. So instead of being all packed up together like a normal shell or a normal sundial, which is the family they belong to, those whorls are separated. So it's a very cool shell. I'll show you a bunch of small turnids, and I had the help of Dr. Harry Lee again to put some of the names on them, because it gets very complicated. We have about 25 species here on Sunnyvale alone of this family. And this is the Bales vitrinella that never reaches beyond 1.8 millimeter. And here is a scale line to give you an idea of the size. This is one of the glassiest, I don't know if I can use that adverb, but it's one of the glassiest shells I've ever seen. It's totally transparent. There's a little sediment here that we couldn't clean, but other than that, it's totally translucent. The terminal vitrinella, very good pictures by James Kelly. And again, a species that never reaches beyond 2.5 millimeters. One of my favorite vitrinellas, a cupola vitrinella, and you can see why it's called a cupola. And the cupola vitrinella never reaches beyond 3 millimeters in size. It's pretty cool. A trilix or trilix vitrinella, 2.2 millimeters. Also very elegant. It has this pentagonal, like 5-side profile to the shell in this area. It is suppressed vitrinella, about 3 millimeters at most. A more opaque shell, kind of milky white in color. One of my favorites, all of them are my favorites. You have to forgive me for that, but this is the fringed vitrinella, because in some cases you can see the periostracum has this spiky fringes sticking off. This one doesn't have a lot of that, and it's a little broken here, but it's a pretty cool shell that never grows beyond 3.5 millimeters. At this point, I want to call attention to this label here. It's a shell that came with the Stingley collection. Dale Stingley, he was a professor at Northwestern University, just outside of Chicago, and he was a very proficient collector and citizen scientist, and he donated his collection in excess of 20,000 lots to the museum at the very beginning, and a lot of them, they make the bulk of, especially the Sunnyvale Captiva collection here. This one was particularly little micro, was collected in 1960 by Dr. Stingley himself. We see more of his collection coming next. Another one of his specimens is the different scale snail, I believe, named by Harry Lee and colleagues from Spain. The names on the label are different than the actual classification we know now. At that point, they didn't realize this was a separate species from the one name on the label. And again, a very translucent and cool looking shell. I have only one Turbonilla, and the reason is this is a group that if you work in molluscologist section in a museum, if you're a citizen scientist or a serious collector, you know how complicated they are, how complex the turbonillas are. Again, it's a group that we have more than 25 species here on Sanibel and Captiva alone. And some of them are probably unnamed species at this point. Harry Lee also let me use some of his images of micro land snails, and those 4 land snail shells come from a place called the Paint Rock River Drift. It's a collecting site called the Paint Rock River Drift in Florida, and none of the species is larger than 1 millimeter. And the genus Punctum is famous for being a very tiny genus of land terrestrial gastropods. I don't have much in the way of bivalves. We're getting closer to the end here, but just want to show you a couple of small bivalve shells. This is a Caribbean corbula from Captiva. And then a shell that's not as small as 5 millimeters, but it's about there. A picture again, image, scanning electron microscope shot by Harry Lee of a Cardiopsis dredged offshore off Jupiter Inlet in Palm Beach County. And that shell is about, I measured here with the scale, it's about 0.9 millimeters, doesn't reach 1 millimeter, or barely gets to 1 millimeter. And you can see a feature here that's pretty cool in this image. You have the adult shell. So this is the fully grown microbivalve. And on the top here, on the beak or umbo of this bivalve, you have the protoconch, or as they call in the bivalve lingo, the protozoconch. So this is the shell that the mollusk had when it was a planktonic tiny bivalve. That's how it works here. This picture taken by John Slapczynski of the Florida Museum of Natural History is a yo-yo clam, and yo-yo clams are super cool because it looks like anything but a clam, right? It looks like a little gastropod. It has little quote unquote tentacles, and the shells themselves are buried within the mantle. You cannot, the shell is totally enveloped by the animal. The animal can, they cannot open the shell and you see the animal inside. The animal is already outside the shell, which is not very unlikely to happen in a bivalve. So they live in the burrows or the tubes of mantis shrimp. So they're commensal with the mantis shrimp. So basically they feed on the stuff that the mantis shrimps left behind. And I think last but not least, I just want to show you a couple of slides from a presentation I did recently at the Florida United Molluscologists group meeting in St. Pete. And it has to do with this very little bivalve that grows inside the shell of our calico, or our common species on Sanibel, calico scallop, although we never found this infestation of the local calico shells by this little bivalve. This is from the east coast of Florida, from Brevard County, and I presented this talk with really cool images done by my colleague John Corbin from the Sandia National Labs in Albuquerque, and John he did the CT scans of that situation, and he colored each individual of the boring bivalve a different color, so we can see that in the presentation. So those are related to angel wings and pidox, and we are still trying to determine whether or not they belong to this species. And I have a colleague now in a museum up in New England that will begin doing, trying to do the DNA sequencing to, to do what we call the barcoding of this species and see how it fits into, the genetics, into the existing barcodes for other species. Here's a pretty cool picture. He did a lot of very cool images. So this is a CT scan, is a method that uses a bunch of different X-rays to make composite images. And you can see there are 13 of those little pidocs. They're called the oyster pidocs. And we really don't know if they are the oyster pidoc. That's the point. Oyster pidocs are much larger than that, but they look like it. And maybe they are, but they just cannot grow much larger because they're growing in a very thin shell. Those shells of calico scallops, they are very thin. Just to give you an example here, I will magnify this section and show you that this thickness here is rarely, beyond a couple of millimeters in thickness. So that may constrain the growth of those bivalves. So I'll keep you posted on results of this small research project. This is how the shell looks like under light photography. They're not very good images. I've done them here at the museum. They're very difficult to photograph. And those are the little bivalves. And in life, because they're protected inside the shell they're boring into, they never close. And you can see here the muscles that shut the shell, the posterior adductor muscles, and a couple of apophyses that they have as expansions of the shell. Last but not least, I want to show you some of the resources we're trying to begin using. And I'll show you a true CT scan in 3 dimensions done by John Corbin in Albuquerque. And this is not a micromollusk, but it's a technique that can easily be used for micromollusks because it gives you a very, very good, resolution and detail. And this being the angulate Weno trap. So this is an image that I can see my cursor here, my mouse. I'm moving the shell back and forth, and you can move it. You can see it from the base. You can see from the apex. So it's a popular thing now. You can use this file to print actually, 3-dimensional models of this little beast here. So with that, I will thank you so much for joining me in this micromollusk adventure today. I want to thank a lot of people here, and I don't have space or maybe memory at this point to thank everyone who has helped beef up the micromollusk collections here at the National Shell Museum, especially the local Sanibel Captiva and Southwest Florida collections. I shall go into this mode here. Harry let me use a bunch of his pictures. Colin made this huge contribution of the Bahamian seashells. Susan Lois, Peter Sharp has given lots of local micromollusks, Herb Chapin in the beginning, Hal Pilcher, Kempiak, and dozens of National Shell Museum donors and volunteers who have made this effort possible of having good representation of micromollusks. So with that, I will thank you, and I'll stop sharing my screen and see if we have questions.
Thank you, Jose. I add my thanks to yours for all the people who have made this collection possible and study of the collection possible and study of this class of mollusks possible. And the photographs, the scanning electron microscope photographs are absolutely amazing. Yours and Harry Lee's and everything, really just beautiful, beautiful images. There are some questions here, and We'll go to the chat and encourage anybody to type in their questions. Ryan had a question about bivalves as micromollusks, which you addressed. He had a couple questions, but he did ask, is there a theory on why bivalve micromollusks are not as prevalent as the gastropods? Or do we just not have as much evidence or as many pictures of them? I don't know.
Well, bivalves traditionally are less than gastropods. I mean, if you look at the different biodiversity diagrams for mollusks, the number of species is about a third, I guess. I can't remember well, and that is reflected into a little into the bivalves. But you have one thing you have to keep in mind, and I mentioned that when I talked about a phenomenon, the general concept of miniaturization, which is what happens when some animal goes from being like 3 inches in size through the evolution of through dozens of millions of years, or vice versa. But there's some changes that are necessitated by the reduction in size. Bivalves, by and large, the largest diversity of bivalves are filter feeders. Most bivalves are filter feeders and not, I mean, by a huge, like in excess of 90%. I guess I'm just kicking numbers. A year later, someone's going to slap my wrist and say, oh, no, it's not 90. But anyway, big number. And filter feeders need to suck water in. And if you're very small, sucking water, I think I mentioned that earlier. If you're very small, let's say you're like hypothesize a 2 millimeter adult bivalve. When that bivalve is sucking water in to filter feed the water, you may find food that's suitable because some microalgae are really small, but the water to that animal has the consistency of thick molasses because of the size. That's a classic physics thing. The larger you are, the easiest it is to suck water in. And if you're very small, it's very complicated. So a lot of those, those bivalves may not be there because of that, because it's very hard to feed. A lot of the very small bivalves are actually, they feed on particles that they grab with their palps, which is very different from filter feeding. So they can do that. Or some of them are predators, which is unusual for bivalves. But and also, generally speaking, there are less bivalves than the gastropods. Gastropods were a very successful branch of mollusks because they evolved in so many different directions and with different habits and feeding habits and all that. And bivalves are not like that.
Thank you. Linda has a question. What percentage of these samples were alive when they were collected? Do you know that?
You're talking about, generally speaking, during my talk today?
That's what I can.
If that's the case, I would say that basically none of them were collected live, except for the ones you saw pictures of that were alive. Yeah, we have them here in alcohol.
Okay. Ellen asks, who discovered micromollusks? Since they are small, how can you tell if they grow and age?
Well, I think when people started getting really curious about, nature and engaging in natural history observations and all that, and I think that one of the things that helped was the invention of, microscopes. People started studying very little, very small organisms like foraminifera, which are little single-cell marine organisms. They started finding those things. I don't know exactly who named or who found the first microgastropod or micromollusk, but I would say it's probably more than 3, about 400 years old.
Okay.
You know.
Yep. Thank you. Helen asked, do they have the same predators that larger species have?
Oh, that's a very good question. Proportionally speaking, yes. But imagine, you know, let's say that there is a kind of fish that eats the crunches or, you know, stingrays here on the islands of Southwest Florida. Stingrays would swallow whole mollusks and crush them with their pharyngeal teeth in the back of the mouth. You may have a little small fish, you know, like 3-inch long fish. I don't know, maybe a blenny or a goby that'll do the same thing with micromollusks. So generally speaking, yes, yeah, there will be diversity there also. You know, if you're too small, then a very large predator won't be interested in you. So they'll have proportionally sized predators.
Mm-hmm. Okay. Two more questions here. Nina asks, are micromollusks more likely to be washed up to a coastline than regular sized mollusks?
I think so. I think that's also a very good question, because you have a lot of you know, there's a lot of sorting, you know, sorting by size, you know, in coastal areas where you have strong waves and all. And if you go to places like and I keep bringing us back to the islands of Sanibel and Captiva, but you go to you know, the Blind Pass beaches, either the Blind Pass beach on the Sanibel side or Turner Beach on Captiva, where you have more stronger waves and more movement in the water. You notice there are fringes of small shells, and if you look carefully, that's where you find a lot of dead micromollusk shells. I see a question by Mark Akin. Hi, Mark. And Mark is asking, are there many naked or shellless micromollusks? Absolutely. Nudibranchs, which are sea slugs without a shell, without shells, they are, most nudibranchs are very, very small, millimeter, 2 millimeters, 3 millimeters in size. And they feed on, you know, a myriad of colonial organisms like hydroids and you know, sea whips and things like that.
Yeah. Okay. Thank you. I think that comes to the end of our questions. And thank you very, very, very much, Jose, for a wonderful presentation. Thank you, everybody, for joining for it. And stay tuned as we open up the registrations for the July and August talks. And thank you for continuing to express your interest in and support for the museum as we rebuild following the hurricane, and we'll look forward to seeing you at a future lecture, or hopefully participating in a future program, and hopefully welcoming you back to the museum as soon as possible. Have a good evening.